Welcome back after lunch. For this session, I would like to invite Dr. Karishma Kaushik. She's going to chair this session and introduce the speakers.
So welcome everyone. It's great to be back after that really good lunch. The lunch was good, but I have to tell you that we have two exciting talks lined up, which is not going to leave much room for a post-lunch nap. All right? So since we're already a little behind schedule, let's get started. Our first speaker, this is the session on modeling the human brain to understand neurological disorders. And our first speaker is Dr. Thomas Hartung. A brief introduction: he's a professor and director at the Center for Alternatives to Animal Testing at Johns Hopkins Bloomberg School of Public Health. He was previously the head of the European Center for Validation of Alternative Methods of the European Commission. He's a biochemist and physician, and he pursues cellular and molecular research into diseases with the aim to ultimately support testing strategies for drugs and chemicals. And the main goal of his work is to move towards a shift in toxicity testing for improved public health. So I welcome you today, Dr. Hartung, and your talk will be on brain organoids to study neurological diseases. You can get started.
Okay, hello everyone. It is just 4:30 here in Baltimore, so I'm not yet at breakfast while you're coming back from lunch. I would have loved to share lunch with you, but like this, I have to come as your dessert or digestive, however you want to see it. Thanks for the nice introduction. You have to allow me to screen share, otherwise I cannot show my presentation. Can you please enable screen sharing?
Yes, they're just working on it. Just a minute.
Oh,
it's done. It's done.
Okay, here we go. Okay. I'm directing Centers for Alternatives to Animal Testing, but I'm, first of all, as you have heard, a physician at the leading School of Public Health. And these marks we actually printed. They read saving more than animals, and that's my strong belief. What we are doing for replacing animal testing is, from my point of view, in the best interest of patients and consumers. I've made my slides available. I will show the QR code at the end of the presentation again, but I also have labeled a couple of papers which I want to make available to you if you're interested with QR codes. So keep your mobile phones ready if you're interested in some of these. Okay. I'm directing a center which just turned 41 actually. So it is the oldest center of the school. There's a team of about 30 people in the US, and we have a European branch now for 13 years. And this European branch is co-directed by Marcel Leist. So we are present on both sides of the Atlantic, but the European team is only about 5 people. These people run very different programs. I'm going to talk only about essentially one of our programs, which is the Microphysiological Systems Program and Developmental Neurotox, which is led by Lena Smanova. But you see here some of the topics we are interested in, from exposomics to developmental immunotox to evidence-based toxicology, green toxicology. So a lot of the aspects what I would consider a modern toxicology, which is more than just testing a product before market launch in some animal tests to say yeah or nay, letting it to the market or not. I see us as a lubricant to make change a bit faster. This is an article we only published 2 weeks ago. It is about the interplay between science, public, and politics. This is where we are around. You might have seen on the previous slide, we are also running policy programs. So we are directly talking to policymakers. We have a lot of activities towards a general public because we want to inform not only to scientists. And this is a very interesting role of trying to make change happen, not just writing the next scientific paper. Our work is possible because we are supported by a number of companies. You can see here some of their logos. But 3 times more money comes actually from philanthropy, which gives us a lot of independence. Though I have to say, when industry is supporting us, we know that we do something meaningful. We know that we do something which is needed, but independence is still a very good, important thing in public health. And our research is funded by the typical sources in the US and Europe, FDA as Food and Drug Administration, the Environmental Protection Agency, the European Commission, the National Institute of Health, and the Department of Defense. And if you're associated with a company, have a rich uncle or whatever, I always find space on this slide. Okay, I also have to declare a couple of personal conflicts of interest. You see that I'm engaged with some companies. Please take anything I'm doing with a grain of salt. I'm not going to talk today about my strong involvement in the field of artificial intelligence, which could be a topic on its own. But you have asked me to talk about brain organoids, which is one of our reputation makers, I would say. If you want to hear more, we made our 2 classes, which we are giving at Hopkins to our students, available on Coursera for free. And you can see in the last 4 years, they have been used by about 16,000 students worldwide. So these are classes, each of them has 13 lectures and some self-examination activities. I really recommend them if you're interested in learning more about modern toxicology and the possibilities. So from my point of view, there's a big societal need to understand diseases, to prevent them and to cure them. And there's only 3 ways to go into disease. One is genetics, and we have seen the Human Genome Project and others over the last 40 years being a strong emphasis of research. But this has by far not explained all diseases. Only about 5% of diseases are purely genetic. We have seen most recently again how different pathogens are. So there's not really a systematic in addressing pathogens as the second line of research, but we do have a lot of factors which are together exposure. And I think exposure science is altogether underrepresented. I believe we need actually a human exposome project, something which is really addressing how the variety of exposures is leading to disease. I've made a living over the last 30 plus years to show that we are not 70-kilogram rats, that we need human models in the end. And if we are honest, human cell culture was not very strong until most recently. It is very difficult. I'm working mainly on the brain. If I ask somebody for some human brain, I will not get it, or at least not as long as the person is alive. And typically, whatever we can get is old, diseased, or whatever. It's not easy to work with human brain, and it's not reproducible as a source. This has changed very much with stem cell technologies. But before moving into this, only one slide on the shortcomings of animal tests. This is a slide put together by my friend Nicole Kleinsteuer, who's heading the US validation body for alternatives. I was heading the European one earlier. And you see here some of the data about reproducibility of different animal studies. And I would say toxicology is the area where we probably have the best reproducible animal tests because they are enshrined sometimes for 40, 50 years in guidelines. They're done under quality assurance of good laboratory practice. They're done just the same by very experienced people. You can imagine these reproducibilities we will not achieve in academic labs with people learning on the job. And with much less strong effects, we use maximum tolerated doses in toxicology. Pharmacology uses subtle doses of substances. So I would say this is a very important take-home message. The value of animal studies is overestimated. We have to see them as one tool in the life sciences, but we also need other tools where we have better reproducibility. And this is leading to effect. Also, the public opinion is changing constantly. We have seen in the US over the last 20 years about 10% increase in people who object to animal testing. We are now at 50-50. About 50% of Americans say they don't want animal testing. About 60% of Europeans say so. And this is leading to legislation. This is leading to some changes. When I'm arguing for new methodologies, then I'm not saying that everything which is cell culture is good. You should see here some of the main arguments. We have an irreproduced cellability. We know that about 25% of academic cell lines are not what we think they are. Very often they are HeLa cells. Very often they're not the species, not the organ, what we thought they are. We still have to fight with tremendous amounts of mycoplasma infection, and a mycoplasma infected cell behaves completely different than a non-infected cell. We see that many cells are cultured too long. They're losing sometimes entire chromosomes. They're completely unstable, multiplying many of their effects. And a lot of the standard culture is full of artifacts. The cell density is only 0.1% of a tissue. The cells are half facing plastic, half cell culture media, and essentially no cell-cell contact. And all this together leads to an enormous problem of cell culture. And there's no game changers. And these game changers are mainly the stem cell technology and the bioengineering, which allow us in more recent time to reproduce organ architecture and organ functionality. And this is called microphysiological systems. And I will give you a bit more on this in a second, and then use our example of brain organoids to illustrate the area. These were two workshops we organized with global experts in this field on microphysiological systems, where we are, where this field is developing. You certainly have heard about the organoids, about the organ-on-chip systems. These are discussed here, and what are the challenges and what is moving forward. If you want a short version, last year, September, we achieved an article in Science on the second workshop. So this is a summary of only 3 pages, much more readable, which is summarizing the state of the art for microphysiological systems. We have also, to be very clear, the role of the FDA in this field of microphysiological systems of critical importance. We in the US are only 4.3% of the world population, but 48% of all money for drugs is spent in the US. And 64% of all money for drugs under patent is spent in the US. And this is why the FDA, which is determining which drug makes it to the market, is what everybody looks for. Yeah? This is the market. This is the agency which is critical for, is a technology acceptable for finding new drugs or not? And this is a very important message you have to understand. And this is why I'm so excited that the FDA is very much engaged in what is called microphysiological systems. Actually, they were the ones to call them microphysiological systems. You see here some slides from my friend Susie Fitzpatrick, one of the opinion leaders in FDA. If you follow their website, you find a lot of materials very different to other technologies. They're not just on the receiving side of things. They are actively pursuing programs and supporting work. Already twice they've funded our own work on microphysiological systems, for example. So as an outcome of the workshop in Berlin, the NIH, National Center for Advancing Translational Sciences here, made money available to organize a world summit on microphysiological systems. I had the privilege to lead in total 52 organizations which teamed up. And you see my 2 co-chairs for the first Microphysiological Systems World Summit, which we held in New Orleans earlier this year. Don Ingber from Harvard WIS and Susie Fitzpatrick from the FDA were the co-host chairs for the first meeting. We had 665 people participating. The FDA was present with 65 people listening in. So you can imagine the enormous interest. So it really shows you that this is a technology which at the moment is looked for. We are going to bring this now to Berlin in June of next year. We will have the second World Summit. There is travel grants available, especially for young speakers. So please stay alert, stay tuned. This is an opportunity. The abstract submission has just opened, and I think it is up until the end of January possible to apply for this. We also formed an international MPS society, which is now developing its bylaws and will be formally implemented at the Berlin meeting. I believe very much in quality assurance. As you have heard, I've handled a validation body. Actually, I started 30 years ago what is called Good Cell Culture Practice, GCCP, because I believe that only by highest standards of quality we can really compete with the quality controlled animal testing, which has good laboratory practice. And I'm also interested at the moment in developing reporting standards for in vitro ongoing work. And actually, we just published earlier this year, and you will hear from David Pames more from this, as he was one of the driving forces of this development. In work over 8 years involving around 100 people, we developed a new guidance document, which is now extending to microphysiological systems. It is a consensus document. We, over the last year of this development, included 350 stakeholder comments, which we received in the document, and I think it is unique in its kind to promote quality assurance here. So now let's talk about brain organoids. Brain organoids, in the beginning we called them mini brains. We have changed our terminology a bit. Work which was originally funded by the NIH, National Center for Advancing Translational Sciences, and early on David Pamies joined us and led these developments. This is an induced pluripotent stem cell derived model, very reproducible in size and composition. We were not the first group to do these brain organoids, the third or fourth, depending on how you count, but we were the first to mass produce them. So we literally produce thousands of these per week. And I will show you a bit of the flavor of these things. What you can see here is the core technology. From induced pluripotent stem cells, we are producing in 2D neuronal precursor cells, neural precursor cells, and then we are putting a cell suspension in such a shaker. And this shaker, here you see, a giratory shaker, allows us by choice of cell density and the rotation velocity to mass produce these little, just like snowflakes, visible brain organoids, which are perfectly round and which are continue to differentiate. Some more recent work addressed one of the unique features of this model, which is myelination. Already in the first description, we showed that 40% of axons are myelinated, and this is something which is pretty rare for cell cultures of the human brain and allows a lot of opportunities for deemed remyelination. I leave it to David to expand on this as you see him. This was in continuation of our work. My interest into the model came from these numbers. And this is the increase in autism in the US. And we see similar numbers in Europe. I have never seen data for India, I have to say. But I would be very much interested. In the 70s, this was a very rare disease. 1 in 10,000 children was diagnosed as autistic. The last numbers in December of 21 were 1 in 44 children in the US is diagnosed with autism. An enormous increase, and nobody knows really what the reason is behind this. But the big question is, are chemicals involved? Because the genetics are not changing so quickly. And we have been interested in following, is development neurotox addressable with a battery of in vitro tests since 2005? when I teamed up between my previous shop, Equam, and my current shop, Cut, and we have been developing a community over time with 4 international conferences and 9 workshops on developmental neurotoxicity. And this is just one of the papers which lays out quite a few of the ideas. Problem here is the animal tests are useless. They cost $1.4 million for one chemical, use 1,400 animals, and essentially they don't find anything. There's no regulatory decisions taken on this basis. And you can imagine how few substances you can actually test. As I said, we teamed up in 2005. These are the conferences. The DNT5 was planned for 2020, will now take place in 2024. And I put here only some of the pertinent workshops. As I said, it's 9 in total, bringing people together. We achieved that the European Food Safety Authority and the OECD took up these activities and are producing at the moment a guidance document on how to combine in vitro tests to assess developmental neurotoxicity. And our own work was funded by the EPA and by the FDA twice. And I will give you of the latter projects a little bit of a taste at least. We have been studying developmental neurotox in brain organoids in the past, addressing drugs like paroxetine and antidepressant, studying pesticides like rotenone or chlorpyrifos and flame retardants. So they are useful. You can actually study these. I would like to address, however, some of the ongoing work to just give you a flavor how this field is exploding in all of its opportunities. The US EPA announced in 2019 that they want to largely move out of animal testing. And we were very lucky that under the leadership of Lena Smirnova and my team, we were the recipients of one of 5 grants the EPA gave out at the time to advance testing. And this was about making a brain organoid-based developmental neurotox test possible. You see the team which teamed up here. So the idea of this project is in our brain organoids, we see a developmental process which is very similar to the formation of the brain and shows especially the early phase, which is very vulnerable to chemicals which produce developmental neurotoxicity. And all of these relevant mechanisms are happening, from differentiation to myelination to neural outgrowth to synaptogenesis, glia migration. And we can also do electrophysiology on these. And the idea of the project is to do this all together. We call this a mini brain bow because we are using fluorescent tags to make the different processes monitorable without destroying the brain organoids. So it is all about high content imaging of these brain organoids and to allow ultimately higher scale drug screening. The central technology is CRISPR-Cas, which allows us modifying genetically our cells and adding one color after the other in order then to differentiate them into brain organoids. This is the first example. What you can see here is a green fluorescent tag behind a PLP1 promoter, which is a marker for oligodendrocytes. So the cells responsible for myelination. And you can see how beautifully you can now monitor these fluorescent cells. And in combination with antibodies against myelin basic protein, for example, you can also distinguish between the early forms and the late mature oligodendrocytes. Here you see the second color added. This is synaptophysin, one of the synaptic markers we are including. So every blue dot is indicative of a synapsis. And we are adding at the moment a second synaptic marker that we have a pre and a post-synaptic marker. We have markers for astrocytes and so on. So this is in development. The second project I would like to share with you is addressing another key hypothesis of our work, which is it is probably a combination of genes and the environment, which is leading to autism. Because we know there is risk genes, clearly, and this explains why we don't have so clear exposure associations. it's not every child developing autism which is exposed, so probably there is a subpopulation which is vulnerable. And in this work here, Lena and team showed the first pair of a risk gene and a risk chemical, in this case CHAD8 as a risk gene and chlorpyrifos, a pesticide which was banned in the US because of its liability for developmental neurotoxicity. And these two are synergistic, as she showed in a paper which was published in 2021, so a year ago, in environmental health perspectives. In this paper, healthy brain organoids were used, and only one gene was changed, a heterozygous knockout for CHAD8, which is the risk factor for autism. And then she was able to show that there is a higher sensitivity towards chlorpyrifos or its active metabolite chlorpyrifos oxone. And this table, without going into any detail, shows you on the left side, ASD are autism spectrum disorder patients. So these are the typical metabolic changes which are reported in the literature in these patients. And you can see something very similar happening if you either knock out CHAD8 or you treat healthy cells with chlorpyrifos or increased in synergy between the two. I think this is a very elegant example of a mechanistic validation where mechanism is used to show that something relevant is happening in these cells, something which is alike to what is happening in patients. And this is something I prefer very much over an animal model in the end of extensive work to show that the key hypothesis is right. We have just been the happy recipients of a large grant, which will now combine this work with 18 centers which are epidemiologically oriented, so that these large studies will address gene-environment interactions in patients, while we are trying to address the same aspect in brain organoids to mechanistically verify what they find, and they will look for specifically these combinations in these patients which we identify. Other work which is financed at the moment by the FDA addresses mixture toxicology. You can see here some work on 4 heavy metals, which all have liabilities towards developmental neurotoxicity. And we're trying to identify how these are interacting and are possibly showing synergistic effects. Another example, just very superficially, recently published again with David, was on Parkinson's disease. This paper shows that what you can do in animal or in a human, which is certain substances wipe out dopaminergic neurons, you can also do in brain organoids. Whether this becomes a better model, I'm not sure, because the animal models have not really led to any drugs for Parkinsonism. But it is at least possible to reproduce what is known in mammals. Another joint paper was on cancer. You can graft brain organoids with glioblastoma, a very aggressive brain cancer. You can see the high density areas of the tumor, which is expanding over time. And then you can test drugs and can demonstrate how this reacts in a natural environment and see at the same time the reaction of the healthy tissue to the same substance. Other work we are doing with the army at the moment is developing glial cells, microglia, the only resident immune cell, which is not part of the brain organoid because it does not develop from the neuronal precursor cells, but it is invading from the York cycle already. As you can see here, we can produce from the same iPSC cells microglia, and it integrates nicely in the brain organoid, which opens up for quite a few studies. This allowed us to study, for example, early on already virus infection, Zika and dengue virus in cells which were including microglia, in this case a cell line, not the iPS-derived ones. More recently, we published on JC virus. Interestingly, this could not be propagated in vitro, but we now know why, because the cells which are being infected are astrocytes and oligodendrocytes, and it was only then in this model where these cells are present that they could be propagated in vitro. We all have gone through a pandemic, hopefully have gone or are going through a pandemic. Early on, we were interested to understand whether the human brain is a target of the SARS-CoV-2 virus. What you can see here was one of the first neurons which we showed to be infected. Each of these red dots is a SARS-CoV-2 virus particle. At the time, this led to a big article in the Financial Times, but 200 other journals followed because we were the first to show that this virus is infecting human neurons. So this was the first peer-reviewed publication, May, June of 2020. So only very few months after the pandemic started, we published this, and a year later, we reviewed the literature. By then, 10 groups had reproduced our findings of infecting brain organoids. And it is all very much in line with the clinic that a subset of patients is showing active infection of the brain. And this might have a lot of clinical consequences. I don't have the time to talk about this. So what you have seen in this
sorry to
project,
interrupt,
yeah?
but if you could wrap up in 5 minutes, we'll have some time for questions then. Thank you.
Absolutely, that's the plan. What I've shown you is here that there is a standardized development from iPSC of these brain organoids possible. You can do this in different genetic backgrounds with risk genes, reporter genes, and so on. We can add glials. We have glial cells present which allow myelination. We can add microglia to make this immune competent. We can study infections, cancer, toxicity, developmental neurotoxicity, as well as neurodegeneration. We have some projects on Alzheimer at the moment and on amyotrophic lateral sclerosis. Gene-environment interactions are possible, mixture toxicology, and we are coming increasingly to functional assays, so not just counting dead cells. The equivalent of an electroencephalogram, and something we call organoid intelligence. And I want to say a few words about this, which is only going to really public in January. This was an early picture when we saw here calcium activity. So every time something is here in real time flashing up, this is a neuron firing. Yeah? And some people said, when I said, oh, they are thinking, they said, oops, are they conscious? And I said, no, they have nothing to think about without input and output. And this led to the idea, which we have been pursuing in a discovery grant with Johns Hopkins, with some colleagues in the Whiting School of Engineering. What we did here is we did develop a type of EEG for our brain organoids, shell electrodes. This is how it looks like. You see here to the right, brain organoid in these self-folding shells. And we just published this in August in Science Advances. And you can now monitor in a much better signal-noise ratio, also pharmacological activities. This is, for example, the reaction to glutamate. So this is a possibility to do something. And we have been expanding this over the last 1.5 years, trying to give the brain now input and output and combine it with artificial intelligence. And what you can see here is another picture of this brain electrodes. This is not new. In 2001 already, Steve Potter started using two-dimensional rat cultures, neuronal cultures. He started to control a small robot, as you can see, and claimed at the time that the robot is learning something and can be controlled. Most recently, our colleagues and collaborators from Cortical Labs published a fascinating paper you might have seen. This was in the press 2-3 weeks ago, where they showed that a culture of two-dimensional culture of human iPSC-derived neurons is playing a computer game Pong. As you can see here, it doesn't matter. You see the pedal is controlled by the neuronal culture, and it is improving and learning how to play this game in a training session. A very fascinating illustration of what can be done here. We see a lot of potential of the three-dimensional brain organoids because it has all of the machinery also for long-term learning. And actually, our friend and collaborator Alison Moultrie in San Diego already a while ago started working on this topic. This is his brain organoids, very similar to ours. And what he is doing is he is controlling through Wi-Fi out of the culture a robot. And you will see the robot in a second. Oops. Oh, the promised robot did not take place. Okay, sorry. That's odd. Did I do a mistake here? Here's the robot. Okay. This is the robot, which he controls through Wi-Fi, as I said, out of a culture. He has not published this in peer-reviewed literature, but already did some pictures were in the New York Times and others. So this is I think, a very impressive way of showing the interplay between machine and organoids. So we're working on this. We have increased our brain organoids to 1 centimeter in hydrogels already, which is twice the size of a mouse brain, which we are using. This has a lot of ethical aspects I cannot really address, but we are working actively with some of the best neuroethicists in this field on this topic. We have organized last February the first community forming workshop on organoid intelligence, which we will publish in January with Frontiers. The Whiting School of Engineering in Hopkins is at the moment promoting this as one of their flagship projects, organoid intelligence. So we hope that they will bring in philanthropic money to make some things happen. So we really want to takes place. Are the biological concepts reflected? That we dream intelligence products. The engineers dream of computing. I dream more of pharmacology and toxicology. And you should just be aware how unique the human brain is and unmatched by computers. The largest computer in the world, the Frontier computer, in June of this year, for the first time, exceeded the computational capacity of one human brain. For the first time, 600 million, 680 square meters of installation. Or another way to say it, if you have an advanced laptop, which is about the weight of the human brain, it is about 100,000 times more efficient, the human brain. And you could pursue Moore's law of doubling capacity every 2 years until you reach a laptop with the capacity of a human brain. 33 years. Okay. We don't know whether we are able to grow a supercomputer, but engineering believes that the future is biological in this sense. I'm interested, as I said, in dementia research, learning. Do we find substances which fuel Alzheimer or make you super intelligent? That's where I see learning systems in the future. With this, I would like to close using economist John Maynard Keynes, who rightly said the difficulty lies not in the new ideas, but in escaping from the old ones. I hope I've shown you some new ideas around microphysiological systems, especially of the brain. The slides are available, and so is our newsletter if you're interested to stay in touch. Thank you very much.
Thank you, Dr. Thomas. We are a little behind time, so we'll take one quick question, especially since it's a virtual talk, and then we'll move on to the next. We do have to start with the front row.
We only talked 36 minutes, or 34.
This is Dr. Subramaniam from Reagent Biosciences. I'm very disturbed by when you said that from 1970s to 2021, the autism rates are increasing, that 1 in 41 children are having autism now. It's very disturbing. I mean, people have been saying that vaccines were the cause of it a while ago. We know, I mean, that may be not true. And you're saying the environment, chlorofluoride, whatever these environmental pollutants are causing it. What do you think the etiology is really going there? I mean, in the developmental neurology, having these birth defects, especially autism, and affecting the neurodevelopment in the embryo?
I mean, first, it is clearly not vaccines. This hypothesis, which has been put forward in an actually fraudulent way, if you go into the history of this, has been disproved in more than 5 million children which have been followed on this. So vaccines are not the reason for this. There is certainly some effects of known substances. For example, lead in gasoline was certainly contributing to some of this, but we have got rid of lead in most parts of the world. There's probably also lifestyle factors. We are getting older, having children. There's effects of obesity, which we cannot exclude. But I would assume that unknown chemicals are making a contribution. This could be substances like flame retardants, could be substances like pesticides, and probably quite a few others which we simply do not suspect at the moment.
I mean, it's a hypothesis that these days pregnancies are becoming later and later. What do you think about it, right? I mean, in olden days...
That's not my field. I'm a toxicologist, but I'm just saying I can very well imagine that this is contributing, but I don't think that there's a single factor which we can identify.
Okay,
Right.
thank you.
Thank you. Next question. Yes, right at the back. We have someone there. Yes, please.
Hi, Dr. Hartung. Thank you for your talk. It was quite enlightening. So I'll just ask a quick question. If you were to pair these brain organoids with other microphysiological systems, could they integrate and take control of them to replicate normal physiology, and how would that work?
Yeah, I mean, the future is certainly in combining organs in multi-organ on chip systems, and we have some projects where we do this. This is not easy, because we typically optimize the conditions for each and every of our organ systems, and then suddenly you have to combine them, and they have different media, different factors. It's also not easy to bring them in close contact, but that's certainly the future. There have already been 2 demonstration projects which have brought together 10 and 12 organs respectively for 1 month. This is high-tech and very difficult. This is probably only 1 lab each which can do this, and they cannot achieve this on a regular basis because you need to have all of the components ready at the right moment. Yeah. But I believe that this is the future to some extent, because we always have to use the most simple system possible to answer our questions, so that we achieve throughput. But if you want to ask how is the liver interacting with the brain, then you should bring them together. Yeah?
Thank you very much.
And any more questions? One more? Okay, we have one in front.
Hello, this is Indumati here. This is again regarding the ASD, autism spectrum disease. So I understand that this is both developmental as well ongoing inflammation related to the disease. So what do you think? Is it reversible by any means once the disease is onset in children? Because a lot of people talk about cell therapy and different lifestyle changes. So is it reversible? What's your take on that?
I'm not working as a clinician. My time in the hospital was very short, and have never treated autism patients. So I cannot tell you what my gut feeling is. I would say that probably damage is done. A lot of this is happening through changes in the forming of synapses very early on. We might be able to improve with some drugs, but in order to find such drugs, you need model systems. And I think that we are coming closer and closer to model autistic situations with brain organoids, and then you can start testing for substance effects. On a metabolic level, we have already shown this, as you have seen. At the moment, we are trying to combine our organoid intelligence approach to show that cognitive development is changing. So we're trying to do OI with autistic children or children with leukodystrophies, which are very similar, purely genetic diseases, for us easier to control, which lead to cognitive impairments. And we want to see how we can use such functional assays. This could open up also for some cures, but I'm not super optimistic that you can heal autism. But you might be able to stimulate some repair, let's say, or do some prevention if you have indications of genetic risks of the parents, if you have indications of risky exposures, you might be able to do some preventive actions during the pregnancy.
Okay, thank you, Dr. Thomas, for your talk. That was a broad sweep on the potential of brain organoids from autism to infections and the mind-boggling possibility of organoid intelligence. So that's left us with a lot to think about. So thank you very much for joining us.
My pleasure. Bye-bye.
Bye-bye.